I, Sapan Shah, Assistant Professor, Priyadarshini Jail College of Pharmacy, Nagpur, welcomes you all. Dear student, today we will going to learn about different techniques of pharmaceutical analysis. Before proceeding to the various techniques of pharmaceutical analysis, let us familiarize ourselves with this term, that is analytical chemistry. So, what is analytical chemistry? It is an application of a process or a series of processes in order to identify or quantify a substance, the component of a solution or a mixture, or the determination of the structure of a chemical compounds. So, right from the starting of a raw material up to the end of a pharmaceutical formulation, you require analysis of that particular substance. If you are going to synthesize any formulation, so you require various excipient that has to be analyzed for its purity, and even after the complete formulation, the formulation has to be analyzed for the percentage content of active ingredient. So it is a very important and integral part in pharmaceutical industries. Now, this analytical chemistry can be classified on the basis of size, like macroanalysis, mesoanalysis, even it is called semi-microanalysis, microanalysis, sub-microanalysis, and ultra-microanalysis when a sample size is less than 0.0001 gram. You can also classify analytical chemistry as a qualitative analysis or quantitative analysis. Now, what is qualitative analysis? Qualitative analysis, it gives the information about atomic and molecular species or the functional group in the sample. Example, the melting point determination, boiling point determination, specific gravity, refractive index, absorbance, optical rotation, viscosity determination, and particle size analysis. All these properties comes under a qualitative analysis. Now, what is a quantitative analysis? It gives the relative amount of one or more of the analyte in a numerical terms. So when you go for determining the percentage purity, the percentage content of active pharmaceutical ingredient, then this type of analytical determination are called as quantitative analysis. Now the analytical methods, specifically the quantitative analytical methods, can be further subclassified as chemical methods, physicochemical methods, microbiological methods, and biological methods. Now the chemical method can be further categorized as volumetric method of determination, gravimetric method of determination, and gasometric analysis. In the B.Pharm first year, you have seen, or if you are studying in a B.Pharm first year, then you will going to learn various volumetric analytical methods. Now the volumetric analytical methods are categorized as neutralization titration, non-aqueous titration, precipitation titration, redox titration, and complexometric titration. So in titration, you will going to measure the volume of titrant required in the chemical reaction. That's why the term volumetric analysis is given here. In gravimetric method of analysis, you will going to analyze the sample on the basis of weight analysis. That's why the term is gravimetric analysis. And gasometric analysis is referring the analysis of various mixtures of gases. Now second is the physicochemical methods. Now the physicochemical methods are various types like potentiometry, conductometry, polarography. It is a spelling mistake, it is a polarography, P-O-L-A-R-O-G-R-A-P-H-Y, polarography, voltammetry, spectrophotometry, colorimetry, atomic absorption spectroscopy, emission spectroscopy, flame photometry, fluorimetry, turbidimetry, nephelometry, refractometry, polarimetry, optical rotatory dispersion, thermal methods of analysis and mass spectrometry. Now you can see these are all various instrumental methods of analysis. So we can call the physico-chemical method of analysis also as a instrumental method of analysis. The instrumental method of analysis or physico-chemical method of analysis, the determination is based on measurement of some physical properties. You can see as per the table, the potentiometer involves the determination of electrical potential. Similarly, the spectrophotometer involves the absorption of radiation, whereas the mass spectrometry involves the principle of determining mass to charge ratio. So in this way, you can classify physico-chemical methods of analysis. Yes, this is a question to all of you. Can you guess this instrument? You have to name the instrument. What is the name of this instrument? If you are guessing correct, then check out. This instrument is called as HPLC. The full form is High Performance Liquid Chromatography, or even it is also called as High Pressure Liquid Chromatography, because it uses a pump to insert or inflow the mobile phase into the column. This is a very important instrument and very widely used in pharmaceutical analysis for the separation and as well as the analysis of various ingredients. The block diagram of HPLC can be represented by this diagram. You can see this is a liquid mobile phase which is present in the reservoir. With the help of a suitable pump, this mobile phase is inserted into via this column. The column contains the stationary phase and the sample. This is a mixture of two or more sample. In this case, you can see it is a mixture of 3 sample. So the mixture of a 3 sample which you want to analyze is to be inserted via sample injector. So mobile phase carries this sample along with it and pass on through the column. The column will going to separate the components, and the component A, which have a very less affinity with the stationary phase of the column, will pass first and will be detected with the help of a detector. The detector will generate a signal, and the height of the peak is depend upon the concentration of analyte which is present in a sample. So if you have a higher concentration of analyte, then the peak size will even higher. If you have a low concentration of analyte, then the peak size will be lower. And the position of the peak is a characteristic of particular sample in a particular conditions. The which conditions? This is a type of a mobile phase, temperature, the column involved, the pump flow rate. All this factor has to be considered for the positioning of this peak, and this positioning we call it as a retention time. So RT is a specific for particular drug in a particular under some particular condition. So this is a very important instrument. In the present day, instrumental analysis is having many advantages over traditional method of analysis. What are these advantages? It is better and faster. It provides more data. As you can see in the previous slide also, the HPLC data, the results are shown in a form of images also. It requires a miniaturization. Now what is this miniaturization? That is, it decreases the sample size. In titration, we require even 1 molar, 0.5 molar samples, whereas in the instrumental method of analysis, even in case of a polarography, the sample size requirement reduced up to 10 raised to power minus 6 molar. With the help of introduction to HPLC, HPTLC, even the sample size has reduced to nanogram and picogram level. That is a preferable advantage with instrumental method of analysis. And last, better data processing methods available with the instrumental method of analysis. Now third, microbiological methods. The microbiological methods are also categorized as quantitative analytical method. Here, one example of microbiological method is given, the method called cylindrical plate method, or even it is called as a cup plate method. Now what we are going to do, we'll take one petri dish. In that petri dish, we're going to keep the bacterial sample. You can keep any strain of the bacteria, and here what we have selected the Staphylococcus. So the bacterial sample is placed in this petri dish is Staphylococcus. Now what we will going to do, we will take a different antibiotic sample and place in the cup or the cylindrical cavity which is placed inside this petri dish. So this is a antibiotic 1, 2, 3, 4, 5 and 6. We will incubate this petri dish for about 24 hours, and after 24 hours of incubation, we will analyze the petri dish for the formation of this zone of inhibition. Now what is this zone of inhibition? The zone of inhibition is representing that this is the area where the Staphylococcus didn't allow to grow. Now this zone of inhibition is nothing but because of the antibiotic sample which is present. So you can see this yellow mark antibiotic, there is no zone of inhibition, whereas this blue mark antibiotic is showing a larger size of zone of inhibition. So what we can conclude out from this diagram is that this blue color antibiotic is having a maximum inhibitory concentration, is the best effective against the Staphylococcus strain as compared to the rest of this antibiotic. So this is a microbiological method of analysis. The second example of the microbiological method of analysis is turbidimetric method of analysis, or even it is called as a tube assay method. In tube assay method, we will take a different test tubes. Like here in this diagram we have taken a 6 test tube, 1, 2, 3, 4, 5, 6, and what we are going to do, we will add bacteria with a nutrient medium in each of this test tube. So we have added the same bacteria in each of these 6 test tube, and then what we are going to do, we will enter a standard antibiotic into each of this test tube. Now the standard antibiotic with a what is the quantity? It is a 1 ml of a different concentration of the standard antibiotic in all. So the standard antibiotic only 1 ml is added in each of the test tube, but with a different concentration. So suppose the first test tube is having a 1% standard antibiotic, second test tube is having 2%, then third is having 5%, fourth is having 10%, and fifth is having 50% standard antibiotic concentration. Okay, so although the quantity is 1 ml in each of the test tube, but the concentration is different. So the first test tube is having a lowest concentration, and this last test tube 6-1 is having a highest concentration of standard antibiotic. We will again incubate this all 6 test tube for the period of 24 hours, and after 24 hours we will going to analyze this test tube, and the property which is measured for analysis of this test tube is turbidimetry. So we'll go for turbidimetry determination of this test tube. Higher the turbidity, it means that the antibiotic has not successfully able to inhibit bacterial growth, and lower the turbidity, it means that your antibiotic has successfully inhibited the bacteria to grow. That is, bacteria are not allowed to grow in the test tube number 6. So this is for the standard antibiotic. Similarly, we can do the same practical for unknown antibiotic sample, and the turbidity value, if same for unknown and as that of the standard, so what we can conclude, the unknown antibiotic is having a same concentration as that of the standard. If same turbidity, it means the same concentration. Likewise. Now fourth one is the biological method of analysis. This biological method of analysis mostly done in the pharmacological labs. So we have taken one example of matching point method, or even it is called as the bracketing point method. Now what we are going to do, let us have one experiment. We have taken one smooth muscle of right intestine, and what we are going to measure, we are going to measure the contraction of that muscle, or we are going to check the effect of histamine on that particular muscle. So you can see the 0.1 ml of the histamine is giving this effect. Okay, so we have taken a 0.1 ml of the standard histamine solution as is representing here as a standard, and the response for the muscle for 0.1 ml of histamine is represented by this peak. For 0.6 ml of the histamine standard, the response is higher. Okay, now you have a test. So what we are going to do, we will take 0.2 ml of the test and find out the response for 0.2 ml of the test solution. The 0.2 ml of the test solution is showing me this response. You can see that this 0.2 ml is lying in between 0.1 ml of the standard histamine to 0.6 ml of the standard histamine response. So what we'll do next, we'll increase this 0.1 ml to 0.2 ml of the standard histamine, and will decrease from 0.6 ml of the histamine standard to 0.4 ml. So we have taken a second response with the 0.2 ml of the standard histamine and 0.4 ml of the standard histamine. The peak size is shown with this height. You can measure this height with the help of a scale. Okay, so 0.2 ml of the standard histamine. Now this is a response for the 0.4 ml of the standard histamine, and this is a response for the 0.2 ml of the test histamine. Now you can see the test histamine is showing the response similar to that of the 0.2 ml of the standard histamine. So we'll again check out with the third analysis. In third analysis, what we're going to do, again we'll keep a 0.2 ml of the standard histamine first response, then second 0.2 ml of the standard histamine response, and third 0.2 ml of the test histamine response. So if all these three responses are showing of same magnitude, we can conclude that the 0.2 ml of the test histamine is having a same concentration as that of the 0.2 ml of the standard histamine. So you have find out the concentration of the histamine in an unknown test sample. We can confirm it with another response with taking 0.4 ml of the standard histamine and 0.4 ml of the test histamine. If again it is showing the same magnitude of the response, so the experiment confirms that the test histamine 0.4 ml, now it is having a same concentration as that of the 0.4 ml of the standard histamine. So this is again a quantitative method of analysis. Limit test is also known as a semi-quantitative method of analysis, where we will not going to determine the exact concentration, but we will find out whether the sample is having a concentration above limit or below limit. So there are different types of the limit tests, like limit test for the chloride, limit test for the sulfate, limit test for the iron, limit test for the arsenic, and so on. All these limit tests are also official in Indian pharmacopoeia, and it is a very important test which has to be carried out in daily quantitative analysis. So the principle of limit test is depend on two things. First is a measurement of the turbidity produced in a sample against a turbidity produced in a standard, or measurement of the color intensity produced in a sample against the measurement of the color intensity produced in a standard. So how you can conclude the limit test? If turbidity produced in a sample is higher as compared to turbidity produced in a standard, we call the sample fails the test, and vice versa. If turbidity produced in a sample is lower as compared to turbidity produced in a standard, we call sample passes the test. And similarly, if intensity of a color produced in a sample is higher as compared to intensity of a color produced in the standard, we call sample fails the test. And vice versa, if the intensity of a color produced in a sample is lower as compared to intensity of a color produced in a standard, we call sample passes the test. So that's all for the limit test. Now let's see what are the importance of the pharmaceutical analysis. The first is the identification of the raw material. As I told you earlier, the pharmaceutical analysis or analysis of the material start at beginning only, that is right from the raw material up to the finished product. Second, the determination of additional impurities can be possible with the quantitative or qualitative analysis. It is also important for the stability of a drug determination. We can find out the strength and concentration of the chemical compound. To determine the molecular weight of chemical compound, suppose you want to determine unknown suppose you want to determine the unknown structure. So what you will going to do, first you will find out the molecular weight, and this is possible with the help of analysis only. The pharmaceutical analysis is having a very important role in pharmaceutical industry as well as in the subject pharmacy. It involves the identification of a raw material, whether it is in process or in a finished product, determination of additional impurities. Maybe your formulation contains a different impurities, you can find out these impurities via pharmaceutical analysis only. Stability of a drug determination, strength and concentration of a chemical compound can be find out with the quantitative or qualitative methods. Even the determination of a molecular weight of an unknown chemical compound, maybe a synthetic origin or plant origin or any other sources. Next is a structure elucidation of the synthetic compound. Concentration of a drug in a plasma or biological fluids can also be determined with various analytical methods. Generally, the mass spectrometry is widely used in such a kind of analysis along with spectrophotometric techniques. Determination of the pKa value, partition coefficient, solubilities and stability of a drug under development. So all this importance suggests that the quantitative analysis is having a very significant role in quality control and quality assurance in pharmaceutical industries. So let us test your knowledge on the basis of some 3 MCQs. This all MCQs based on the previous video only. The first question to all of you: which one of the following is a quantitative analytical method? Options are viscosity determination, pH measurement, refractive index, titration. So pause this video for 1 minute, answer the question and then move ahead. If your answer is titration, then yes, you are correct. So question number 2. All of the following are electrochemical method of analysis except first conductometry, second potentiometry, third amperometry and fourth refractometry. So in this case you have to select an option which is not an electrochemical method of analysis. So again pause the video as per your required time and then move ahead. The correct answer is refractometry, as it is involve measurement of the refractive index, and it is called as a qualitative analytical method, not a quantitative analytical method. Question number 3 is quantitative determination principle in titrimetric method are based on. So first option, on the basis of measurement of volume, on the basis of absorbance of sample, on the basis of pharmacological response, and fourth, on the basis of measurement of conductance. So take your time, pause the video and then move ahead. The correct answer is the quantitative determination principle in titrimetric method are based on measurement of volume. So thank you all for watching this video. I hope this is a very informative session to all of you, and I have tried to incorporate most of the analytical method which you require in pharmaceutical industries for quality control and quality assurance department. Thank you very much.